All right, so this is kind of a talk that I was just talking to Pete today. This is a talk that could probably easily be a whole course in controversies, mysteries, and challenges. And this is sort of right kind of in one of my wheelhouses as far as, kind of what I think about a lot, and so you'll probably, I'll definitely end by 3 because I have to end by 3. But there's a lot of information in this, and stop me if you want any clarification with anything. And certainly most of the slides, at least a lot of them, have references and things like that. And of course, you can have copies of the slides and things like that. So just to begin, these are my ChatGPT generated images of controversies, mysteries, and challenges in fMRI. And I'd like to usually start this out with sort of the way I think about fMRI method development, or fMRI in general, is being organized across 4 pillars of fMRI, what I typically call pulse sequences and hardware, paradigm design and processing, interpretation and contrast mechanisms and physiology, sort of understanding the nature of the bold signal, the neurovascular coupling and things like that. And then finally, basic research and clinical applications. And over the years, so actually, and sort of if you really want to look at how these break down, I mean, everything from, increasing the speed, resolution, sensitivity, robustness of the pulse sequences, looking at, everything from similarities and differences across subjects for paradigm design and processing and methodology to, how to normalize across subjects, how to capture the relevant neural activity and behavior with various paradigm designs. And then, of course, as we do with resting state, do network modeling of that. This all is contained in that. And it's, of course, in interpretation of contrast mechanisms, we're trying to understand the various shapes and structures of the dynamics of the response. Of course, all the neural activity that goes into the hemodynamic response, and how it relates also to non-neural physiology, the artifactual responses. And of course, for applications, certainly we'd like to develop biomarkers. It's currently used for pre-surgical mapping. There's things like neurofeedback platforms and things like that might be useful clinically. And also we want to use it to fundamentally understand the brain. And we'd like to, and when I say understand the brain, I mean more than just how fMRI is used 99% of the time today, which is sort of mapping and looking at how those areas change in activation magnitude. And that, I'm sort of trying to push along the lines of going from that to sort of causal mechanisms, actually trying to understand, how the brain is actually working, as opposed to pointing to areas and say, this part of the brain is active, therefore we understand something. And we actually don't really understand that much more just by looking at and mapping. And so, there's ways of going about that. Of course, you can plug in your fMRI data into computational models, for instance. That's one level of trying to understand mechanisms. But you can try to go deeper if it gives hints to deeper models that then can be looked at with other modalities. So a few of the significant, I don't know why that particular one is, a few significant advances of fMRI over the years is, let me just move this. So for instance, ever since fMRI began, in terms of the hardware, we've developed multi-channel RF. We had a couple of clever innovations in imaging acquisition that everyone uses today, the ability to do simultaneous multi-slice that can essentially shorten your TR for getting a whole volume of brain slices, and also parallel imaging techniques using the RF coils to help spatially encode the data. That was a major advance, going to higher field, certainly an advance. Going to sequences that are sensitive to more capillary effects, like vaso, is a big advance. Multi-echo, as you heard about a couple of weeks ago, or maybe last week, that's an advance in itself. Being able to, people have used that for 30 years, but it's only starting to become more common as far as being used. And going to higher resolution. Our resolution has continuously been increasing. Not that much, though. Right at the very beginning of fMRI, people used 3.5 millimeter voxels. They might be using 2.5 now. In general practice today, you can go much higher, though. I'm not going to go into all the paradigm. I mean, lots of advances in paradigms and processing, everything from the collection of big data sets that people can analyze. That's a huge advance in itself. Going back further, the establishment of standardized platforms was critical. You know, and there's other advances too that actually this area, paradigms and processing, has probably been, for the last 20 years, the most active area in fMRI by far. And that's, if you go to OHBM, you'll see most methodology work along these lines. And interpretation, it's been sort of going along slowly. There's maybe a handful, maybe a little bit more than that, groups actually doing animal models in which they're doing calcium imaging, single unit electrodes, optogenetics, where they're really probing the neural response simultaneously with fMRI to try to look at the relationships. And there's been some mostly confirmatory information, but some differences sometimes are seen, which is opens for future study. And there's been lots of behavioral work where it's tightly sort of related to the fMRI signal changes. And then certainly applications. The way I look at it is that fMRI is unique in the sense that it lends itself to whole brain imaging. It's non-invasive on humans. So whole brain maps that are correlated with behavior, maps and networks that are correlated with behavior has been the major application for most of our research. And for applications as far as clinical, it's only been pre-surgical mapping, and that's only a tiny, tiny fraction of clinical work, and probably a fraction of what the potential of fMRI is. So, okay, what has been a true impact of fMRI? So resting state correlations was massive paradigm shift in understanding the brain. Other methods have been able to look at this, but it never really gelled in neuroscience that whole brain networks were correlated. So fMRI sort of opened up that window. It's not just an fMRI thing, it sort of opened it up for neuroscience. There's obviously more delineation, better delineation of sensory processing. There's been new functional structures discovered, looking at the hierarchy, especially in visual cortex. Lots of advances that were unique to fMRI. Identification of changes with learning, adaptation, and plasticity. That was really hard to do before fMRI came along. So now we can. Identifying multiple brain networks in resting state tasks in association with behavioral state and traits. This is also something that really wasn't done that much. I mean, a little bit with EEG, a little bit with PET. It was really hard. So that sort of exploded. We have a much better perspective of how the brain is organized at this macroscopic level. Once again, though, does it give us insight into how the brain is actually computing? Not really, but it gets us closer. And actually, it's funny how fMRI, there's been a lot of work on neurovascular coupling, but fMRI really catalyzed this work to look at, how is it that the vasculature, changes its flow with neural activity. So that's been a lot of work. But as I mentioned, there's not much clinical impact, many suggestive clinical research findings, but not much impact. And fMRI hasn't been completely informative as to causal mechanisms. So it's still in the realm of sort of cartography. We're sort of mapping out these areas and seeing how the areas change with time and with behavior, but we don't really, or we're not really getting, deep insights as to how the brain is actually working and why it's organized as it is, So for instance, we know what the frontal lobe kind of does, but we don't know why it's there. We don't know, all the connections that it has and what it's actually doing. So, but this is where fMRI lends itself to neuroscience in terms of the temporal and spatial scales. Spatially, it can span across many different scales, all the way down from circuitry, all the way up to whole brain information. Temporally, it really captures a huge swath of what's going on in the brain, all the way down to milliseconds, tens of milliseconds. You can have an event with fMRI that's down to 16 milliseconds. It's like maybe it's about the shortest event that I know of that you can still elicit a response. And so you can't necessarily discern two parts of the brain that are delayed by 16 milliseconds. You can't even do that much lower than 100 milliseconds at best, and typically about 2 seconds. But you can look at very brief activation. So, and that's nice. And so, and the reason why is because, most neuroscientists, though, would argue that this is still missing everything. That if you really want to understand the brain, you have to understand it at this level, at the level of molecules and microcircuits. And that probably is not true. You probably have to understand it across these scales. But if you really want to know mechanisms of computation, then we're right at that edge. We're right at that edge. Maybe a little bit too coarse still, but at the level of columns and layers, you might be able to get at something. Okay, so now let's start with the main part of my talk. I'll hopefully spend about 15 minutes with each. So starting with controversies. So fMRI has sort of been defined over the years. It's been sort of fun to look at all these controversies, and many of them have touched upon people here at the NIH, for better or for worse, and they sort of help catalyze the field. So let's start with things that are controversial controversies, because then people may not agree with me on this, but this is my own opinion, by the way. Practices that may be incorrect. For instance, the use of, and hopefully a lot of these are, if they haven't been taught yet, there's like maybe a pointer to somewhere in the course that they will be mentioned, or you can always look it up in the literature. Practices that may be incorrect. I mean, people, when they were trying to clean up the signal, they thought, oh, I'll take the global signal of the whole brain and regress it out. And that cleans it up. It certainly does. But it also changes the relative magnitudes of signal changes over time, because a lot of the brain has that signal. And if you suddenly regress out something that is a big fraction of the brain, you're distorting all the increases and decreases. So that's something that's been debated. One of my postdocs, Kevin Murphy, wrote sort of the definitive paper pushing against this. And it's still debated. It's still being debated. I think the general idea is that it shouldn't be used, but it sure does help in certain contexts. Connectivity interpretation. So this is my feeling, is that when people look at resting state correlation, it's resting state correlation. And so if they see a correlation change either across subjects or with a task, that's a correlation change. To call it a connectivity change is taking a massive leap. That's probably completely incorrect. It might be lucky and be correct sometimes, but there's many things that can change correlation that have nothing to do with connectivity. You can change the noise in the time series from one or the other nodes that show correlation. Their connectivity isn't changed really. Just because they're in sync also doesn't necessarily mean that they're connected. I mean, they're sort of they might be driven by some other source. And then also, if your model function, if your regressor doesn't account for phase delays or lags in the signal, you might have two signals that go in and out of phase, and they might look like they're more or less connected, but they're actually just going in and out of phase, or the shape of the hemodynamic response might vary between the two, and they might show that they're not as correlated, but they might be perfectly correlated. So my feeling is that it's an important point to me, and it might sound esoteric, but to not call this connectivity, just call it correlation. Because I don't even know what connectivity means. I mean, think about it. I mean, usually it means, if two areas are, have a cable connecting them, and somehow increased connectivity is like the bandwidth is increased or something. And there's no equivalent of that in terms of what we're doing. So it's just correlation of the signal. But that said, it works pretty well. And actually you might find areas that are more in sync with across subjects or different populations or with a task. And so it works well. And so it might be an esoteric thing that I say. It might actually end up working. Parcellations. So typically, and I'll go into this a lot more as I go in my talk, but typically with resting state, looking at resting state correlations, people first parcellate the brain. They have a standard parcellation template divided into like 500 parcellations, and then they analyze the time series. They can do network modeling on the time series. And the problem is, though, is that every brain is unique. And while we have similar parcellations, they're not identical. And people try to normalize brains into a parcellation template, and you're probably throwing around maybe a third of your data just by doing that, or mixing it in a way that reduces your sensitivity. So parcellation's a tough one too. Thresholding. Gang Cheng, who's a statistician here, will probably be talking about this. Picking the threshold is a big thing that we all have to do, but how we pick it makes a big difference in terms of how comparable our results are, how much of a statement we can make about them. And I'll give a few examples of how it might not be a good idea to the threshold at all. Simple regressor models. When we typically design an experiment, we have the experimental timing, and then we usually take a boxcar or event related and convolve it with the hemodynamic response, and that's our regression function, and that's our model function. But the reality is that the brain is doing probably many other different things that are time locked still, but don't look at all like that function or don't have the same phase. So when we put in one model function and we say, oh, well, there's activation and there's nowhere else activation, we're probably wrong. There's probably activation in a lot of other places. We're just missing it because we haven't modeled it. So, all right, a lot of, discoveries of shortcomings that got amplified by non-experts. And even though I have a bunch of slides about these in detail, I'm realizing I'm going to just quickly go over this sort of stuff. So findings that are intriguing but have not been widely reproduced. For instance, many people have seen with maybe ends of 10 to 500 results of depression or Alzheimer's or different neurologic disorders that are suggestive, but they're not conclusive. There's also been other things like neural current imaging or looking at direct neural activity, that papers that have been put out there like once every few years. They generate some amount of excitement, but then they're never replicated and they disappear. So that happens a lot, especially with things like just recently there was a paper that claimed to see neural activation, not replicated. It made its way into science as well, unfortunately. Or fortunately, it helps catalyze the field to look harder at these things. Activation-induced bold and white matter. Several groups, and it's sort of growing. Several groups have seen bold contrast in white matter. We haven't, but they have. The pre-undershoot, it's another thing that, typically with an fMRI response, when you give a stimulus, the signal goes up, and then there's a big post-undershoot that everyone sees after it goes back down. But a few groups early on claimed to see a pre-undershoot, thought that was very significant because it shows like maybe metabolic rate increasing before the flow kicks in, pulling the oxygenation down. Hasn't really been replicated that much, more than neural current imaging, but not that much. And maybe comparable to white matter imaging. So it's one of those things that not enough groups are working on it to really definitively nail whether it's there or not. And so it's kind of an interesting thing. As I started with many small n group comparisons. So looking at these comparisons, like, so for instance, this made a big splash back in 2009 by Surratt and Das. They were doing this electrophysiology study, and they were giving stimuli. And every once in a while, they would not give the stimuli. They would give it in a regular interval, and then they wouldn't give it. And what they saw was a sort of a hemodynamic response, what they claimed, without any neural activity associated with it. They're like, oh, look at this. fMRI is wrong because we see a hemodynamic response, but no neural activity. And so everyone sort of, the media, they didn't say that, but the media would say, oh, okay, fMRI is wrong. It turns out that it's more complicated than to interpret it this way. Dan Hammerker and I wrote a paper sort of pushing back against this, saying, this is probably due to the fact that your stimuli were close together, so there's still residual oscillations of the bold response that are overlapping. And also, if you really look hard at the neural activity, you can probably see some, whereas the bold signal is more sensitive than the electrophysiologic measures. And so it could just be a difference in sensitivity. All right. This one was actually useful to the field. The problem of circular analysis and double dipping. And this was a really good catch by, it was actually a postdoc of mine and here in LB Laboratory of Brain and Cognition, Nikos Kragaskordi, and among other people. And they realized that a big practice that people do without really even realizing it is that a lot of times you want to do correlating, like for instance, a behavior or response with the signal. And so using the same data set, you might select areas that show a correlation with what you're looking for. And so you find your regions of interest. And then you use those and look at the response from those average regions of interest from the same data to look at the effect. And you're like, oh my gosh, this is like 100% correlation. And then you report that. And there's other gradations of that sort of circular reasoning that can occur that, and it's all laid out in this paper. And they identified about 40% of the papers that were being published had these unnaturally high correlations with behavior because there was this pre-selective filtering of the data based on some bias or some way of using the behavioral to select it. And then they were then looking at some result based on that. So you can't do that. So you have to probably keep your data sets separate is the safest way to go. There's other ways of going about that, but that's beyond the scope of this. Just be aware of this paper and be aware of this effect because it's really important. And most reviewers catch it now. This one was sort of fun, but it was kind of useless because it was something that everyone, this was an example of something that literally everyone doing fMRI knows, that you could have false positives. And if you don't do the appropriate corrections like Bonferroni correction, whatever, you'll get false positives. I mean, it's obvious to everyone. And this person submitted to OHBM, not really to make a statement, but actually as a joke poster, practically. It won the Ig Nobel Prize, one of the Ig Nobel Prizes in 2012. And so the media picked this up and they loved it. They're like, oh, look, we asked dead salmon how they felt about these images, and we got activation in the brain. And, the literature picked it up, amplified it. Everyone's like, oh my gosh, fMRI is, giving dead salmon brain activation, we can't believe it, which was sort of funny to everyone. It was never really taken that seriously by anyone in the field. But it's a good example of false positives. This one was more recent. This was the clustering conundrum. And the idea here is a lot of paradigms, a lot of post-processing algorithms, what they do instead of looking at each voxel independently, which they're not, they look at them as grouped, as having spatial dependence, but they also try to cluster them based on, they try to bootstrap up the signal based on their proximity. A perfectly legitimate thing to do. And this is an interesting example of something that looks on the surface like a massive error. This paper came out that showed, so this line down here shows what the false positive rate should be, like 5%. And this is across processing platforms, including fMRI. And Bob Cox, when he was here, who wrote this clustering algorithm, they even in the paper said, oh, there was a bug in fMRI. On top of fMRI being wrong, there was a bug. And it really upset Bob, like more than anything else in his entire career. So, and the reason why is because, kind of irked him that they would point this out without first talking to him. They could have talked to all these people and just worked it out without publishing a paper. But they published the paper, and they claimed that, that up to 60%, I don't have the fraction here, but some huge fraction of papers that use this, the results are completely wrong, which is absolutely not true. So when you have an area of activation, the cluster might be bigger by a few pixels, or there might be other small clusters around. I'd be willing to bet that if you actually go back to those papers that all have done this wrong, and you have a little bit more salt and pepper here and there, maybe the clusters are slightly bigger, did that ever change the results, the conclusions of the paper? And the answer, I would argue, is almost 100% no. So even though it was a huge problem, it's important to correct. The media picked up on this. Actually, Tom Nichols got a lot of pushback from the field by writing a blog post saying, 30,000 papers are wrong. and which they are. But in fact, if you really want to argue that way, every fMRI paper is wrong. I mean, it's like saying, a common analogy is like saying when you fly to if we're flying to San Francisco, the plane is off course all the time, and so, yeah, they're wrong, but at the same time, it didn't make that much of a difference. It's important to correct, but it didn't make that much of a difference. All right, and more recently, there's a couple of papers that came out. There's one paper in which 70 groups analyzed a single data set. And they all had they had agreed upon 9 hypotheses that they were supposed to test. And it turns out that the conclusions of the paper, as written, were that the results varied all over the place. But if you actually look at the paper, and this is what happens often, is that if you have a result based, you analyze the data, you have a model regressor, you threshold, and then you determine the spatial correlation of some highly threshold result with a cognitive task, so it's not that strong of an activation, with other groups, and you have a threshold result, yeah, they're not going to look that correlated. It's going to look not very correlated. But if you lower the threshold, and that's this red diagram right here, if you actually have no threshold, just look at the patterns of activation everywhere, they're pretty highly correlated everywhere. So the results could be skewed to be, they're all different, depending on whether you have a threshold, or they're all the same, except for these unfortunate groups that completely screwed everything up and got the paradigm backwards. So that's the blue correlations. But aside from those people, if you do it generally right, then everything kind of is close. But that brings to a head the idea that the results are naturally skewed in 2 ways. One, when you apply a threshold, and then when you interpret your results as saying either the activation is there or it's not there, or we get a result or we don't get a result. So most of fMRI, most of the world, in reality, is not like that. So it's sort of like, well, there's results that seem like they're weighted towards this direction, and this is the trend. This is what we see. So it kind of was an eye-opener as far as that's concerned. And it did sort of foster the idea that having open data sets that you share and compare analysis is a good thing. It's always a good thing. All right. The last example is a recent paper by Merrick et al. Really, really nice paper that really catalyzed the field. It was in 2020 that kind of had a really depressing message, though. Basically saying that in order to say anything, when you're doing a group comparison, let's say you have a group of people with psychosis and you have a group of people who are normal. And if you're putting them in and doing resting state analysis, parcellating, putting them into a standard template, resting state analysis, how powerful is that paradigm? What results can you get? Well, it turns out if you're trying to see any difference, he argued in his paper that you need like 2,000, approaching 2,000 subjects. Otherwise, all of your data are artificially enhanced in terms of the significance, which is a real message, and it's actually true in this context. And then everyone sort of started beating their heads as to against the wall as to what could be causing this. Well, and it sort of brought to light a couple of things. One is that people are all different. So even the groups that had psychosis, there's many, many, many different types of psychosis. They're all pooled together. And this also brings to a head the issue of the parcellation templates. So if you parcellate a person's brain, their parcellation pattern will kind of look the same. It'll be close, but not exact, relative to another person's brain. If you try to put them into a same template, you lose information. And so when you then look at resting state averages and see how the correlations are different between certain people, those and others, of course you're going to be giving up power. And also doing a univariate analysis is much less powerful than trying to do multivariate analysis on the data. So they had a lot of good messages. Everyone realized, okay, they're probably right in the way that fMRI is done right now. And it's important to note, it's not a criticism or a statement about fMRI sensitivity, because you can get clear activation with a single subject in a single run. So fMRI is plenty sensitive to activation. And it's very sensitive to differences in activation. You can tap your fingers 1 hertz, tap it 5 hertz, you'll see a difference. So it's very sensitive. Just the way that people are pooling the data across, selecting their populations, the paradigms they're using, how they're putting it into a normalization template, not accounting for all the individual variation, that's what's causing the lack of sensitivity. So if that could be fixed, which is very doable, then you can really improve things. All right, so what are some of the mysteries? All right, that was just a few of them. There's other controversies too that I could talk about, but so mysteries, at least mysteries that, once again, this is biased to me. And if we run out of time, maybe I can give this as a supplementary and another talk a little bit later. Okay, so one mystery is why do we get a bold response in the first place? Another one is, what are the mechanisms behind the bold transients, nonlinearities, and inhomogeneities? Can we see inhibition versus excitation? What are the sources and types of spontaneous fluctuations in connectivity? That story is getting more and more complicated today. What is the global signal? This is one that I've been thinking about a lot. There's some really good papers on this. What's the purpose of resting state? We don't even know what the purpose is. We've been using it constantly. We don't even know if it's an epiphenomenon or actually does something useful. What are we missing in activation? Like I mentioned, simple regressors, we generally throw away a lot of information. And this is my thing about parcellation. Why is the parcellation size the size that it is? How did we arrive at that? And is it better to do finer parcellations? What's the most meaningful way to compare subjects with parcellations? Do they vary across individuals? Do they vary over time? What's the best way to compare brains across and within groups and across individuals at different resolutions? So for instance, a big mystery, it's kind of more of a challenge, but you have layer fMRI, super high resolution fMRI. There's no way that you could put that into any standardized template right now and be able to make a statement as to, to be able to have enough accuracy to average multiple subjects together. So how do you do that? Nobody knows right now. Everyone has sort of ad hoc methods of doing that. We're just kind of in the early days, the way fMRI was, 30 years ago, when it comes to layer fMRI and how we deal with this data at this high resolution. How relevant is the spatial and temporal scale of MRI for deriving principles of brain function or cognition? So let's just start with the first one really quickly. And I'll try to go as fast as I can in the next 5 minutes for this section. I thought this argument was settled. I actually thought that everyone says the standard thing that people say is that, oh, well, you have increased blood flow to deliver more oxygen to the tissue because it's more metabolically active. And so there you go. There was a recent paper that came out right here. Dinozzo is the first author, basically saying that that may not be a driving factor. The argument is that you had to have more oxygen in the blood because the delivery of oxygen was diffusion limited in the tissue, and so it had to be like, because it's an exponential, you had to fall off of oxygenation to give enough oxygen to that furthest neuron that's active. You have to overshoot the oxygen in the blood, and that kind of makes sense, and everyone agreed about it. But this paper came out saying no, tissue neurons can work just fine with whether it's 90% oxygen or 50% oxygen or 30% oxygen. They're functioning just fine. What the real reason is it's to maintain pH, eliminate CO2, and eliminate other sort of waste products of metabolism. If that waste product is there, there's a lot of damage caused to the vasculature and to the neurons. So it's really bad for that. So it's important. It's much more important to get rid of that waste than it is necessarily to deliver the oxygen. So, and they have basically some pretty invalid models describing this that suddenly put this whole thing back in question again. Another mystery. So what are all the mechanisms behind the transients? I'm not going to go into too much detail, but basically, there's a lot of nonlinearities in the bold response that we still don't understand. This was a classic paper by Logothetis showing that if you varied continuously the contrast, like checkerboard contrast, the bold response varied in some linear way, the local field potentials varied in some linear way, and so it was linearly coupled. But it turns out that he didn't really talk about this part, is that when you if you go from like hardly any contrast to zero contrast, it's suddenly very nonlinear. So what this is saying is that the bold signal overestimates very, very weak stimuli. So if you have a very weak stimuli, you'll get a larger hemodynamic response than a linear system would predict. And then it becomes linear after a while. And this is another example of this. This is some work I did with that Rasmus Byrne did when he was in my group, basically showing that with very brief stimuli, up to about 2 seconds long, the hemodynamic response way overshoots what a linear system would have. We don't understand why this is. Is it a nonlinearity in the neural response? Is there some nonlinearity in the hemodynamic coupling? We don't know. And a more recent example is the work by the MGH group at Martino Center, basically showing that if you drive the signal really rapidly, like 1 second on, 1 second off, so basically almost 1 hertz, or 1 cycle per second, you still are able to elicit a hemodynamic response, which is confusing because by any linear estimation, that should be totally in the noise at that point because it's slow. And their model for that is something that looks like this. They have, you see my cursor? Yeah, okay, I can't even see my own cursor. They have like this dotted line right here that says, okay, when you're driving the signal really fast, the response gets faster and it returns a baseline faster. So we don't know why that happens exactly. Could it be some blood volume effect? or what? We don't know. And there's all kinds of mysteries that have popped up in the literature. This is some work that I did a long time ago, still hasn't really been explored that much. Like when you compare different types of visual stimuli, this is a flashing diffuse red, you get a response, but you don't get much of a post-undershoot. If you get alternate black and white pinwheel, you get a nice large undershoot. This is looking at the same area. We don't know why that is. We don't fully even understand what the post-undershoot does. Here's another more physics-like example of modulating the post-undershoot. If you apply what's called diffusion weighting, getting rid of the intravascular signal in the blood, you can reduce the magnitude of the response because most of your signal is gone. It's disappeared from the intravascular part, but that doesn't seem to affect the post-undershoot magnitude at all. And that's a mystery. All right, along the lines of post-undershoot, there's been a couple of nice pieces of work looking at trying to look at inhibition and excitation. One was where in an animal model they applied GABA. GABA is inhibitory, and so there's more of an inhibitory action on the gold signal. And they find that it certainly modulates it by decreasing it, maybe having more pronounced transients at the beginning, and then more of a post-undershoot. Sanji Kim actually also, using optogenetics, this is a plot on the right here, selectively activated inhibitory versus excitatory neurons and found hugely different responses. Excitatory is mostly like that black response. The inhibitory neurons caused this transient and a massive post-undershoot. So that gave me hope when I saw that. It makes me think, okay, maybe we can figure out the relative degree of excitation and inhibition in each voxel based on trying to tease apart the signal into these sort of two components. It's a thought. So that's a mystery. What's the sources and types of spontaneous fluctuations? So there's lots of work on this. Everything from spontaneous neural activity to respiration to thermal noise and so on. And just looking briefly at some of the fluctuations. This is work by Javier Gonzalez Castillo in the group, basically showing that if you take a seed voxel in the fourth ventricle, which is sort of CSF, and you find that as a subject gets more and more drowsy, this signal becomes more prominent, as you see with this power diagram right here, frequency versus time. And you find that this low frequency fluctuations, 0.05 hertz, get higher by a lot as people get drowsy and fall asleep. If you use that as a regressor, you find this really interesting effect, and both in sleep and during the awake state, that the base of the brain, supposedly CSF, but it probably contains some bold signal, shows this slow traveling wave that goes across the brain that's anti-correlated with cortex. So the subcortex is anti-correlated with the cortex. What exactly this means, we don't know. But there was a suggestive paper that was by Logothetis in monkeys, where they had electrodes on the hippocampus during sleep, and found that when you used a regressor against the hippocampus, the same pattern appeared, that the base of the brain, all the subcortical areas were anti-correlated with the cortex if you regressed against the hippocampus. So it feels like we're close to some sort of insight here, but we haven't really gotten it yet. Another type of oscillation is one sort of pioneered by Shelley-Kielotz, looking at quasi-periodic patterns. And that is, once again, it's not the 0.1 or 0.2 oscillations that we typically see in resting state. It's lower frequency. And she found like there's these three predominant patterns in the brain that seem to show these slowish oscillations that are not respiration, and they're not global signal, but they're these slow oscillating signals that seem to divide in the same way that the brain is sort of loosely organized. So what do they mean? I don't know. Another way of looking at resting state that is sort of sort of opens up or helps explain a mystery is that when you think of resting state fluctuations, most people think oh, it's 0.2 hertz, and you're picturing a sine wave. That, oh, it's just a bunch of sine waves that are 0.2 hertz, nicely oscillatory. Turns out, Josh Faskowitz, right here in the room, has pioneered this method looking at edge analysis. And what that essentially is it's like taking a sliding window, but with a window width of 1 TR. So every time point is multiplied against the other time points. So pairs of time points are multiplied together to get, if it's a high value, there's a high, it's basically, you get the precise moments in which those time series are highly correlated. And what he found with this, I mean, if you can, it explodes the data out. But what he found, and you can put these in matrix form as well, is that a couple of things. One, and that's basically an example of what you do, is that you have this one time series, another time series, you multiply them together, you get something like this. And so when they're correlated, it shows high here. One of the many things he's found is that these matrices vary a lot over time. But one thing that I just want to talk about, though, is that when you calculate the correlation coefficient of the time series by just looking at every voxel correlated against every other voxel, you can get a map something like this. And that's basically is mathematically equivalent to just simply averaging up the edge time series. So, but if you actually, it turns out that if you look at these edge time series, and if you average across all the edges at every point in time, and this is the time series right here, what he found is that it doesn't look like this. This definitely doesn't look like a sine wave. It looks like, or neither is it sort of steady state signal. It's a bunch of these bursts or spikes, as he calls them. And it turns out that if you take the data and you average up all the spikes, you can get a correlation map that looks like this. But if you only take the 3% of the data, just looking at these spikes, average up just those, you get the same map with about the same statistical power, pretty much, which strongly suggests that resting state correlations, resting state oscillations are not oscillations. In fact, they're just events that occur every once in a while. And so that kind of redefines how we think about it. And taking this a little bit further, if you just count up the spikes, and this is sort of recent data, if you just count them up, it turns out that the spike count per parcel seems to match 1-to-1 with the average correlation that you find from one time series to another. So it's all just a function of number of spikes in the duty cycle. All right, so what's the purpose of resting state fMRI? I feel like I'm gonna get to not very far into the challenges, but oh well, we'll see how it goes here. I really do have to end at 3 though. And I'll continue this. Maybe we'll have a supplementary talk. So what's the purpose of resting state fMRI? This is old data that we collected that was intriguing, that I sort of stared at now for 15 years. I've been staring at this. And what it is essentially motor cortex. And if you look at it, and if you take a seed voxel along the motor cortex, you can see, as expected, that there's a high correlation of the corresponding opposite side of the sensory motor cortex area. So as you move that seed voxel along, the other side shows a correlation. But there's some ripples in that. And so taking that a little bit further, you can see if you divide it up into, I'd like to get my pointer back, but oh well. Anyway, that's okay, I'll make do. If you take this and divide it into foot, hand, tongue area, and look at the magnitude of the correlation as you go across between the left and right motor cortex, you find that there's a clear dip in the hand area where there's a low level of correlation. And so I thought about that. I thought, well, maybe resting state has something to do with, how synchronized the brain is. It's very sensitive to how synchronized the various parts of the brain are. So you can imagine when you're walking, your legs are always moving kind of in sync. they're always both moving. When you're talking, or the face part, the left and right part of your face is always in sync. But hands are much more rarely in sync. They're all or doing something else. So they're still in sync, but they're less in sync. And so I thought, well, maybe this might be a measure of just how generally synchronized the activity is during the course of the day or the course of the week. And there was a really nice paper that came out a couple of years ago by Nico Dosenbach. Actually, Nuvo Dosenbach's on it. It's from the WashU group. And essentially, they measured the left and right motor cortex correlation, and then they had really motivated volunteers, and they put their arm in a cast, and they had them wear this cast for 2 weeks. And within 1 day of putting on that cast, the left and right motor cortex suddenly showed much less correlation. And then they wore it throughout the entire time. There was more to this paper, but basically the idea is that when you took off the task, it went back up. So to me, this indicates that resting state correlation is very sensitive, not to some global intrinsic state of the brain, but kind of what it was doing yesterday. And it's very dynamic, it seems. So that's actually an insight into resting state that I think sort of suggests as to what its purpose might be. And there was a really nice paper by Pizzulo, written in 2021, and basically they mull over this whole thing. They have one quote. This is a nice diagram from Josh as well, but it's just kind of showing this oscillating brain. But basically, they're saying that that this low dimensionality of these spontaneous neural patterns, they might reflect, well, not only the low dimensionality of our behavior, but slow transitions between generic priors tuned for low dimensional general purpose behavioral patterns. So basically it's tuning the network to what it kind of just did to sort of make it sort of optimized for what it just did. So it's a network that's sort of tuning itself. So the itinerant dynamic sequences of the brain states may reflect exploration of the brain's dynamic repertoire to remain flexible. So it's something that occurs that sort of optimizes the network activity. So I kind of like that. It's not an answer, it's not a solution, kind of opens up sort of attempts at network modeling of resting state that could be interesting. All right, so what are we missing in activation? That's another mystery. And it's an open mystery because we don't really usually know what we're missing until we look. So here's one. I thought this was one of the most beautiful examples that really hit the point that everyone should really read this paper by Camille Uludog, basically showing that everyone knows about the classic hemifield checkerboard hemifield experiment. You shine a hemifield on one side, you get hemisphere on one side, you get activation on one hemifield. Shine on the other side, you get activation on the other hemifield or hemisphere. So it switches, and that's everyone thinks, okay, then we're done. We have regressors that look like each one, and you get different hemifield activations. Well, it turns out he did one other thing. He put in one extra regressor. He put a regressor that showed a transient at the beginning and a transient signal at the end, a brief signal at the beginning, a brief signal at the end. And what he found was a very different map so what you see on the left is your classic hemifield map, just one side being activated by the opposite side. But with the transient activation map, you get this bilateral activation that is very different than here. So it's clear activation that shows up that anyone doing standard modeling of the paradigm would completely miss. And I think that this is everywhere in our data, everywhere. And this is another beautiful example that illustrates this point. This is some work by Javier Gonzalez-Castillo, in which he averaged subjects together by 9 hours of averaging. So he got super high signal to noise data. And instead of using any model to try to capture this simple paradigm, the paradigm was a simple blocks of design where subjects were looking at a checkerboard, and there was either a letter or a number within that checkerboard. They just simply had to tap a button when they saw it. And you'd think, okay, you'll get motor cortex, visual cortex. That's it. What he found is that if he just had an open-ended model and did k-means clustering on the shapes of the data, it turns out almost 100% of the gray matter was activated. And it was activated in a way that clearly shows similarity to how we understand the networks of the brain are organized. And each of these time series looks like just noise. I mean, except for like here and here, this is motor and visual cortex. It shows some big deactivations. It shows some weird, the double transient right here, all kinds of everything in between, but it's all real. It's all super significant, and it's organized in a way that's organized by network in the brain. So even when you're doing a simple task, every single part of the brain is active, which is both cool, but also kind of a little bit scary in some sense, because if all the brain's active for everything you do, then what are we doing? And trying to, the key is then to try to tease apart what's more active or less active if we get to that high sensitivity. So I'll just finish the mysteries in this talk, and then I'll leave the challenges to another talk. But in this particular talk, actually, let me just go through these last 2 or 3 points here. Okay, why is the parcellation the size it is? So typically, when I say parcellation, when we look at resting state analysis, we either do seed-based sort of analysis, which is sort of exploratory, or we put the brain into a standard parcellation template to do matrix analysis or other types of analysis on it, like any sort of circle plots or brain template plots or things like that. Turns out there's many different parcellation templates that are out there in literature. I count 16 here, but there's more. And they all use various mathematical approaches to get at them. And sometimes we use these, people like the Yao atlas, or they like the Schaefer atlas, or whatever. But it turns out that if you look at the internal homogeneity of the signal within each parcel, you find that the atlases vary a lot. It turns out that, and this so happens to be Evan Gordon from WashU. They have a really nice parcellation technique that looks at resting state correlations and where there's boundaries in which areas are correlated. And that seems like it finds areas that are highly homogeneous. But problem is that even if you make those areas smaller and smaller, things get more and more homogeneous. So why does it make sense to keep the parcel size the size that it is? Why don't we try to keep on making it smaller and smaller? Of course, there's a signal to noise issue, but if there's not, is there some principle by how the brain is organized such that it naturally falls into these parcels? That's kind of a mystery in that regard. And there's evidence that it's obviously organized in much finer detail. And this is just one example looking at resting state with motor cortex activation, where you iteratively find ICA components, you find one component, then look at the ICA of that, and then look at the ICA of that component. And if you work through enough iterations of that, you can get down to individual digits that show unique ICA components with resting state. And there's other evidence that these parcels vary across subjects. And this is where I started with this talk, with one of the other controversies. Parcels vary across subject, and there's less variation in the primary sensory cortex, but a lot of variation overall. And it seems that it's not random how they vary. It seems like the parcellation is extremely tied in spatially with where the myelin is, or the myelin landmarks in each person's brain. So if you can map their myelin, you can help guide where the parcellation should be. So that might account for the subject variability. It might be a way to normalize. And then Todd Constable has a paper out where he even shows that the homogeneity of the signal, how well correlated it is with itself, varies depending on the task. So even with the different tasks, you might have it might not make sense to think in terms of parcellations because they're subdivided depending on what task is going on. Okay, it's 3:05. And I have my challenges, which is another 20 minutes. So I'm not gonna, I'll save this. I promise I'll save this for maybe a talk next week. I'll fit it in one of these times here. So with that, I'd like to thank you. Thanks.